Okay, welcome everybody to the number 3 online webinar. Thank you so much for tuning in to this important industry webinar tonight, and welcome you from the team at the National Varroa Transition to Management Program. I'm your host, Bianca Jiggins, from the Australian Honeybee Industry Council. We're the peak industry body for honeybee industry in Australia. I'd like to acknowledge the First Nations people as the traditional custodians of the lands and waters which we live and work. I pay my respects to elders past, present and emerging. This evening's program is coming to you from a number of different angles. We've done some research in the background, following up from the Public Information's team's collation of several key questions that have come in from both the audience last time and what we gather throughout the entire T2M program. And as a result, this presentation is predominantly around topics to do with Varroa biology and a short update as well. So Tamara Prentice, the National Transition to Management Coordinator, will bring you a short overview of where we're up to with the program. And then we're moving into a deep dive into Varroa 101 or Varroa biology from Dr. Madeline Kratz from the New South Wales DPI, the Honeybee Industry Development Officer. We'll have a short Q&A session following that. So please chuck your questions into the Q&A down the bottom of your screen. Just a quick couple of housekeeping points before we go forward. Don't hesitate to put Q&A function questions in, but we will answer throughout when we can, and we'll hold as many of them as possible to the end, just to make sure that they're part of the live Q&A and to have them all completed into that final recording. It's especially important as the final recording will be added to YouTube for future reference. The Q&A will be a valuable resource for the whole audience. This webinar is being recorded, so please, and it will be published online, so it's important to remember that the information presented tonight is current as of the 2nd of July 2024. And if you are watching the recording of this webinar, then please make sure you check with the websites for up-to-date information. We'll move on to introduce Tamara Prentice, our first speaker for this evening. And Tamara's going to give us a quick update on where the T2M program is up to. Thanks, Tamara.
Thanks, Bianca. Hopefully you should be able to see my screen now. So good evening. As Bianca mentioned, my name's Tamara Prentice and I'm the National Program Coordinator for the Transition to Management Program. Tonight, I'm hoping to give you an update on some of the things happening at the program level before we hand over to Madeline for the details on Varroa 101. So what's been happening? So since the last webinar at the end of May, the program's really started to pick up steam. We held our second Train the Trainer. This one was down in Melbourne. It was cold and it got dark really early for those that haven't been recently. So Train the Trainer is where each of the jurisdiction's trainers and Varroa development officers get 2 days of intensive Varroa training. These trainers get in-depth consultation opportunities with a variety of experts in the field so they can answer whatever questions you might throw at them at future training events. If you're interested in becoming a trainer, Queensland and WA trainer contracts are about to be advertised. So watch this space from the 4th to the 18th of July if you are keen to become one of our trainers. The training for beekeepers has also continued with the first South Australian course to be run in Clare on the 20th of July. So we're a bit excited about that one. The New South Wales training is recommencing on the 12th of July. So this will be the last run of training in New South Wales. So there'll be quite a few training courses, but this will be the last lot available in New South Wales. So if you can get along, please get along before the New South Wales training is finalised. Some of that training is up on the border at Tweed Heads. So if there's any Queenslanders on the computer tonight, you're more than welcome to come along, get a head start before it gets to Queensland and join us at one of those training events. Vic and Tassie as well, your training is starting to be proposed in late July and August and running on from there. So you're not too far behind in your training either. We've taken on some early feedback from the earlier run of New South Wales training. So some of the recreation courses now you'll see are being offered on weekends, and there's also specific commercial training for commercial beekeepers. We are continually aiming to improve the program. So if you have any feedback for us, please send it through. It can only improve the future training for your future beekeeper friends. Other project activities are also continuing in the background, including the scaling down from the initial Varroa response. Another thing that might be of interest to the people in the crowd tonight is that we've got a request in at the moment to extend this program until February 2026 within the same budget envelope. Currently, the program finishes in 2025, in February 2025, except for Varroa development officers. The intention is that we can support some training being available for beekeepers in the winter or the cool season of 2025 for those states that haven't been able to complete their allocated training schedules this year. That's not all states. So if the training is near you now, please don't miss out on the program because it is time limited. We're not guaranteed that extension. And some states like New South Wales, for example, the training will be finished in 2024. The national program staff are trying to get our message out as broadly as possible around what the program is up to and what resources are available. We'll share a link to subscribe to the weekly update in the chat, and that's where you can get the most up-to-date information on a weekly basis so you don't miss out on opportunities near you. The program's also acutely aware that pollination is nearly upon us. We have a position in the program for a pollination industry coordinator. We're actually holding interviews for that position this Friday. That position's going to be super busy, preparing a pollination industry guide and helping pollination reliant industries understand the impacts on their businesses as Varroa spreads. Victoria's borders are now open under a permit system, enabling bees from around the country, including from New South Wales, to enter. The conditions are pretty tight, including, amongst other things, that the apiaries must never have had a detection of Varroa, and that those hives must also have had a recent inspection by someone who has been trained by an approved training course and found to be free of Varroa. There's been some questions asked about why Vic are opening the borders to enable the New South Wales bees in. So I just thought I'd touch on that briefly. So almonds are worth about $750 million a year in exports. They need about 300,000 hives for pollination each year, and that's just going up as more almonds are planted. Between New South Wales, SA, Queensland and Vic, there's around 560,000 registered commercial hives. So over half of these need to move to almonds. A lot of these beekeepers rely on the pollination contracts to support their businesses. We know there's a risk, so do the jurisdictional governments. And so we're asking people to be prepared and to undertake any training opportunities that are available. And we are trying to tailor those to meet where some of the beekeepers will be during that pollination event. The Varroa development officers. The Varroa development officers are employed to provide on-site assistance to beekeepers. They're actually overseen by Bianca. So any questions about VDOs, Bianca will be very able and capable of answering. So in New South Wales, the VDOs are already on the ground. They've also just recently started in Vic as well, as you can see on the screen. But in New South Wales, they're able to see what's happening on the ground as the mite spreads. And I've included a couple of photos from this week near Nowra, just to show you what they're seeing. On that bottom photo, you can see the Varroa excrement. It's the white in these cells, and it indicates that there's a massive mite load. In the top photo, you can see a stack of dead mites that were trying to escape a brood cell. The hive itself was dead. The bees had absconded, probably with a massive mite load, and those bees that were remaining were dead and had symptoms or showed to be consistent with parasitic mite syndrome. Madeline's going to talk more about this, but the VDOs are seeing hives dead within the 16-week recommended testing interval. So you'll note that the recommended testing interval is now being reduced to 4 weeks where Varroa is present to account for these massive infestation or reinfestation rates while the mite populations are unstable. We're learning as we go from what our VDOs are seeing on the ground and from each other. In Australia, across the climates, we're going to see different treatment needs, and the VDOs are a great resource to find out what is happening and what's working in your area. However, as I mentioned before, the program is time limited, and there's some chance that the program will finish before Varroa even gets to your area. So once the VDOs are on board, get in and learn how to deal with this thing before the time runs out. The VDOs also get a bigger reach when they're attending club and association meetings. So if you're holding an event like that, reach out to your local VDO contacts and they can get along and help your club. At the last webinar, I mentioned there was some cool data in the background of the Varroa heat map. So the cool data is where negative surveillance has been reported, and hot data is where positives have been reported. Since the last webinar, we've published a new heat map, and you can see that on the screen. It lets people see where both positive and negative surveillance results have been reported, and it also lets us filter by time frame. Some areas showing Varroa were euthanized, and the Varroa was eliminated as best we know. So limiting the map to a more recent time frame can be more accurate in your region if you are one of those areas. You can also check whether recent monitoring has been reported in an area if you are deciding to move your bees. This is the style of map that's proposed to be made national, subject to approval by the National Management Group. Each of the jurisdictions will share de-identified data to enable the map to be collated across the country. The message, however, tonight is the same as last time. The map's only as accurate as the data reported. As an example, the photos in the previous slide followed a very recent positive report. The large mite counts indicated that Varroa had probably been present for quite a few months and hadn't been reported. So those beekeepers either weren't undertaking the surveillance or weren't reporting what they were finding. And so we just really would please encourage you to report your surveillance, either positive or negative, so that we can target training and education in those areas as the mite spreads. I wanted to end my presentation on a little bit of a positive note. Varroa is a problem, and we're all well aware of this. We're starting to see some positives, not on the scale of Varroa free, don't get me wrong, that would be bliss, but we're not seeing that. We're currently happy to take small mercies. We have decades of overseas experience we can draw from, so we're not starting from scratch. We can modify our hive practices to suit our climate and deal with Varroa alongside the small hive beetle we've been dealing with. We have a range of chemicals here to choose from as well. Though they're not specifically designed for Australian beekeeping conditions, we can adapt and find solutions. We do that here. We can learn from overseas experience with resistance. We have a unique opportunity to avoid repeating the same mistakes. Unlike New Zealand, where only one chemical was available, we have multiple options and multiple modes of action. And I'm sure Madeline will focus a lot on that tonight as well. Well, actually, probably not, sorry, that's a training thing. So those beekeepers who have survived with suboptimal management of American foul brood won't cause similar problems with Varroa reinfestation. If their management practices don't improve, their hives just simply won't survive. There's no longer an ability to avoid inspecting brood nests. Ultimately, the industry will benefit from higher standards and improved beekeeping practices. Varroa will compel beekeepers to collaborate regionally. We'll need to share information to understand what's happening in our own areas, to coordinate treatments and to prevent reinfestations. This doesn't mean revealing details about good sites or honey flows, but it does mean working together effectively. Feral colonies will largely disappear, reducing competition for floral resources. There'll also be an increased demand for replacement queens. So this will prompt an increase in queen prices and enable those producers to deliver better products. Leveraging decades of selection expertise, we'll be able to prioritise Varroa mites in our selection criteria. One last plug, at the moment, there's support available for the beekeeping industry and pollination dependent growers. Please leverage this support and take advantage of the program offerings while they're here. And that's it from me. Now I just need to work out how to unshare.
Thanks, Tamara. We will cross over to Madeline Kratz to provide us with a wonderful presentation on Varroa biology. Take it away, Madeline.
Thanks, Bianca. I will just share my presentation as well. Can everyone see that? Perfect. Well, thanks for having me tonight. And I'm Madeline Kratz. I work with the Department of Primary Industries as a honeybee industry development officer. And as already mentioned, today I'll be talking about Varroa biology and impacts on honeybees. I would like to start off with, let's see, one second. Oh, there we go. By acknowledging that Varroa destructor is in fact a parasite of honeybees. So by definition, that means that a parasite is an organism that lives in or on an organism of another species, its hosts, in this case honeybees, and benefits by deriving nutrients at the other's expense. So in other words, that means that in fact, honeybees have no benefit whatsoever by that relationship with the mites, but it is entirely the mite that is benefiting in this instance. And the reproduction of the mice is also entirely dependent on the development of honeybee larvae. And in fact, that is also why, in this case, so honeybees and Varroa mites have a very closely linked relationship in terms of the lifespan of honeybees, and that's what allows Varroa to replicate and reproduce, and which prevents Varroa to, for example, reproduce with other honeybee species, like, for example, some of our natives. I just want to dive a little bit into the history of mites and how that is relevant to us today. So the Varroa originally, the original host, or the parasite Varroa, was originally linked with the Asian honeybee Apis cerana. And Varroa, so there's different species of Varroa, but Varroa tecumsehana was originally identified on the Asian honeybee in Java in 1904. And it was by the 1960s that Varroa had already jumped species and could be found on Apis mellifera, so our European honeybee, and that was in the Philippines, in Japan, and Russia. So you can see on the map how Varroa over time has spread. And unfortunately, that global spread was mostly linked to our human activities in terms of us introducing honeybees to different countries and different continents. So by the 1960s, 1970s, Varroa had spread to places of Europe, to other regions of Africa, also North and South America. And it wasn't until early 2000s that it was actually discovered that the mite that was affecting Apis mellifera was in fact a different species to the mite that is affecting Apis cerana, and that's when Varroa destructor was named. One other thing that I would like to point out here is that, in fact, one big difference between the Varroa affecting the original host, or the Asian honeybee, compared to Varroa affecting the European honeybee, is that where Varroa actually reproduces, because in the Asian honeybee, Varroa can almost entirely only reproduce in drone brood, which in Asian honeybees gives them a huge advantage in terms of being able to cope with the mite. I mean, they have evolved with Varroa for a much longer period of time, but it also is a seasonal advantage that the Asian honeybee has in terms of mite numbers being able to reproduce only when drone brood is around. Unfortunately for our European honeybees, and also for beekeepers keeping European honeybees, Varroa destructor on Apis mellifera can reproduce in both drone and worker brood, which makes it much more challenging in terms of managing Varroa in our European honeybees. Now that brings me to the life cycle of Varroa. So most of you, or some of you for sure, have already heard about the two phases that Varroa has in terms of its life cycle. So there is what we call phoretic phase, which means that is the time where the Varroa is in fact present on adult honeybees, and there is a reproductive phase where Varroa reproduces. So during the phoretic phase, Varroa like to attach themselves to nurse bees, and the reason for that is that the nurse bees are the bees that are looking after and actively feeding larvae. So for that reason, the mite can jump from the nurse bee into the cell and starts reproducing. And that is also the reason why when we do our monitoring and our alcohol washes, sugar shakes, or soapy water washes, why we really want to get down into the brood nest and collect our 300 bee samples from the brood nest, ideally from brood frames that are open and capped, so that we can get a representative sample of the mite load of that colony. Now, so once the mite travels with the nurse bee, then drops into the cell of the right age. It will immerse itself into the larval food. In worker brood, that happens 15 to 20 hours before that cell is sealed off. In drone brood, that happens from around 40 to 50 hours before capping. That mite will then pierce a hole into the developing pupae, which then becomes the feeding hole for not only the mother mite, or the foundress mite as it is also called, but also for any other offspring. And the mite will then start laying its first egg on the side of the wall, which will turn into a male mite. And any consecutive eggs that the foundress mite will lay at 30-hour intervals then turn into females. So the males will then mate with the females as they mature, and mate development from egg to adult takes about 7 to 8 days. And one critical difference between mites reproducing in worker brood compared to drone brood is that generally speaking, you can get 1 to 2 mites emerge from worker cells, whereas 2 to 3 mites will emerge from drone brood cells along with the mother mite. And that is because drone broods take a few extra days to develop, 24 compared to 21 in workers, just allowing the mite to have more mature females hatch from that cell. So in general, once the daughter cells emerge with the mother mite, they then undergo another phoretic phase. So they're back on the nurse bees, on the adult worker bees for a few days, and then they can restart the whole cycle again. So one female mite can go through 2 to 3 cycles over the course of the lifespan to have that chance to reproduce. The male mite, on the other hand, and any females that are underdeveloped generally will die in that cell. The lifespan of mites, this is also interesting, is between 28 days to 5 months. And you might think, oh, that's actually a pretty long period of time that the mite could survive for. But I would like to think of it in terms of our summer and winter bees. So if you have mites that are actively reproducing during spring, during summer, they will have a shorter lifespan compared to mites that are surviving on winter bees or colonies that have gone broodless, that are surviving on adult bees, and they have to be able to get through to spring. So they can live for several months in that instance. The other interesting fact, mites are actually blind. So that means that they entirely rely on pheromones and chemical cues to be able to detect nurse bees and to find that right-aged larvae. So there's chemical cues that the brood itself gives off, and that also the larval food releases, that the mites can pick up, and that will tell them, okay, this cell is ready for me to jump into and start reproducing. On that note, mites also prefer to breed in drone brood, and we know that in drone brood is 8 times more infected than worker brood in Apis mellifera carnica colonies, for example. And that has to do, well, for one, we know that the development stage of drones is longer, but there must also be chemical cues that tell the mite that this is a drone brood compared to a worker brood, because they cannot physically tell the difference, if you like. The other thing that you might observe when you start drone uncapping some of those cells is that you might see some drone brood that is not affected by mites at all, compared to other pupae that have several mites on the same larvae. And there's some theory that that might have to do with aggregation, but there could also be chemical cues again that make potentially one larvae more attractive to the mites than another one. So we've been talking a lot about mite reproducing and mites feeding, but what do they actually eat? So Varroa mite mostly feed on fat bodies, and that happens during the reproductive phase, and it also happens during the phoretic phase. And I just want to spend a little bit of time first looking at what are fat bodies, because we often talk about them, but might not necessarily familiarize ourselves with where exactly they're found on bees. So this is a cross-section of the bee abdomen that you can see on the right of this picture. And within that picture on the left, so the middle section that we're looking at, you can see a fat body of a very well-fed bees or nurse bees or wintering bees that would have good nutrient stores to either be able to overwinter or to be able to feed larvae, for example, so nurse bees. And for that reason, it also allows Varroa to feed on that tissue. So on the right, on the very right side, you can see what fat bodies that you would find in forages, for example. There would be far less, because they don't have the necessity to feed larvae, for example, or in colonies that experience malnutrition, they would also have underdeveloped fat bodies. Now, mites and this is also why mites are found on bees in certain locations, because they do like to tuck themselves into that third segment on the underside of the abdomen. So that gives them the biggest pocket, if you like, to protect themselves from grooming of the bees. But it is also where the highest concentration of fat bodies is found on honeybees, which is the perfect feeding location for mites. Now, in terms of the function of fat bodies for honeybees, if we compare it in human terms, it is like our human liver. So it is involved in nutrient storage, but also in detoxification, in water regulation, hormone regulation, and in immunity. So it really plays a crucial role for honeybees. Now, what happens if Varroa actually feed on honeybee larvae and pupae? What sort of symptoms do we see in pupae or in adult workers that develop from parasitized pupae? Now, we can see that there's definitely a decrease in body weight in adults. Many of you might have seen photos of adult bees with shriveled wings, and quite often we link that with deformed wing virus. But in fact, deformities in bees or in adult bees can also show up from Varroa simply feeding on pupae. Another sign is that adult bees have been found to have smaller hypopharyngeal and mandibular glands. So they are the glands that specifically nurse bees rely on to be able to produce larval food. Loss of body protein makes sense. Fat bodies store nutrients for honeybees, including protein, which they store in the form of vitellogenin. And this is also becoming the location where mites pierce the pupae or the adult bee is also becoming the site for viruses to enter and affect the health of honeybees. In drones, people have also found reduced emergence rate of drones and also effects on, on drone physiology, so specifically effects on fertility and reduced sperm count. On adult honeybees, the impacts of Varroa are on immunity. Again, fat bodies play a crucial role in that. We see a reduced lifespan, reduction in foraging, the tolerance to pesticides is reduced, and again, this is another opportunity for viruses to also introduce viruses. Now, I want to spend a little bit more time talking about viruses, because viruses can be passed on to parasitized pupae and adult bees, but once the viruses are present, like if you imagine how many bees are inside of a colony, there's also viruses being spread between bees. which can be through contact, which can also happen through larval feeding. And in fact, there are, globally speaking, there are more than 20 viruses that affect honeybees. So this is an example or a picture of deformed wing virus in honeybees, which, as the term suggests, affects the wings of honeybees. They shrivel up, they cannot fly anymore. But at this point in time, deformed wing virus has not been detected in Australia. We are on the lookout. So there's still active testing of viruses happening in the background. But there have also been other viruses previously detected in Australia, and I just want to mention those as well. So the viruses that you can see here that are listed in yellow have previously been detected in Australia. They, through a study that was run at CSIRO, John Roberts has done in 2015. And viruses such as sackbrood, black wing cell virus, Kashmir bee virus, Israeli acute paralysis virus, chronic bee paralysis virus, and Leishmania virus type 1 and 2 have all been previously detected within Australia. There's still other viruses such as slow paralysis virus, deformed wing virus, and acute bee paralysis virus that we currently do not have in Australia that are having a big impact on honeybees globally. But all I would like to say, talking about viruses here, is that in fact there are plenty of other viruses that we still have that mites can now act as vectors for to spread, and we might be able to see the impacts of these more so on colony health. So the ones that I've marked with an asterisk on this slide, we know that that mites can certainly act as vectors for those viruses to be spread between bees and between colonies. Now, the other thing to note about viruses, like any other pest or disease that we might see in honeybees, virus levels vary with season, but also from year to year. So just like chalkbrood might be more prevalent in one year or small hive beetle in other states, the same thing happens with viruses, where they might pop up more in one season or more in one year compared to another. Now, speaking about the effects on the colony, so the effects of Varroa on colonies is not as simple as we would probably want it to be, but it really depends on a lot of different factors. So right now, there's in some areas where beekeepers are seeing really high infestations, but maybe not quite the effects on the bees, and then suddenly the colony completely collapses. So it really depends on infestation levels, also on reinfestation of the environment and of Varroa colonies, but also on secondary infestations of other diseases. So Varroa, for example, already weakens the immune system of honeybees, and then other diseases such as chalkbrood, nosema, and other funguses and bacteria are much more likely to impact honeybees. And it's similar to us humans. If we are constantly exposed to viruses and bacteria, but doesn't mean that we are always being sick. But if our immune system is down, we're much more impacted by whatever is around. Now, it also depends on the climate. In some cases, there might maybe be a brood break or very slight brood break. Most of Australia, probably not. Also food sources. Are the bees nutritionally supported, or are they experiencing another stressor by not having the nutrients that they need? And I just want to spend a little bit more time on nutrition, because it is often something that sort of hides in the background of people's minds, but it can be a significant stressor for honeybees. And even now with Varroa, it is more so something that we need to pay attention to. So honeybees do have essential nutrient requirements. And aside from carbohydrates, which they get from honey or nectar, pollen is the major source of nutrients for proteins, amino acids, fats, minerals, and vitamins. And pollen can vary largely in the nutrients that they provide. So depending on the plant species, there might be a better source of pollen in terms of nutrients, and also provide more or less quantity of pollen. Now, this image is of pollen that I've actually trapped very recently. And you can see it's mostly of one color. Orange is a very good chance that it belongs to Asteraceae family, which is the daisy family. And that is pollen such as flatweed, fireweed, dandelions, and it is in fact a very poor source of pollen. So we really want to understand, what are the bees foraging on? What are they getting? Are they getting the right quality and quantity of nutrients that they need? Because pollen protein can vary between 7 to 35%. So about 25% is considered good or average, but anything less is a very poor pollen source, which in this case, daisy family is usually around 15, 17, so not a very good source for protein. Also, some essential amino acids or fatty acids can be lacking in different pollens. One study that has looked at supporting colonies nutritionally, or actually have been several studies, sorry, but this is one example, where they have had colonies on an apiary, and they divided those colonies into two different groups. So one group was nutritionally supported, the other group was not. All of the hives were infected with Varroa, and interestingly, but probably not surprising, there were similar virus levels in those colonies, but the colonies that had the extra pollen given to them showed improved tolerance and resilience to viral infections. So the viruses did not have the impact, or not as strong of an impact, than the colonies that were suffering nutritionally. They also found that adult longevity was increased when they had the extra pollen source, and they had increased capacity to rear brood, and they also performed much better throughout winter. So they experienced far less colony losses when they provided that extra pollen. At the end of the day, the only way to really reduce viruses is controlling Varroa mite. But the key message here is that well-fed bees are more vigorous bees, and more vigorous bees handle Varroa better. With that, let's just have a very quick look at how the dynamics between population increase in honeybees and Varroa mites behave. So some of you might be very familiar with this graph. This graph is based on 4 seasons, but the big takeaway here, I believe, is that as the colony naturally increases over spring and summer, reaches its peak, there will become a critical time where Varroa is still trying to catch up, is still reproducing, numbers are still building up, and then there will be a time where the colony naturally decreases again. So that would be in our autumn, but the Varroa levels are still high. And that is a critical point for not only monitoring, which should happen throughout the year, but also in terms of managing those mite numbers before the colonies go into winter, to make sure that they are as low as possible, and that you can start off with a colony that is managed in springtime, rather than ending up with a mite explosion in spring as such. So that is the critical thing to keep in mind. So active monitoring is a must, and it is critical to determine Varroa early, to understand what mite numbers are we talking about. And the only way that we can do that, as I said before, mites are very easily hidden away in the abdomen of bees. Visually, we cannot tell what the mite levels are. If you can see mites visually in your hives, that hive will not be around for much longer. So we really have to rely on our alcohol washes, sugar shakes, and soapy water washes to keep on monitoring mite levels. In terms of monitoring, so according to the biosecurity order, it has been 4 times per year, at least every 16 weeks. That is currently changing, but best practice from now on will definitely be monthly monitoring, whether we're already dealing with mites or whether you're trying to find mites early if you're not affected with mites just yet. And that brings me to the treatment thresholds. So the treatment thresholds are still best practice to keep monitoring, but also treating Varroa once you reach a threshold of 5 mites between 1st of March and the 30th of November for each year. And the same goes for 9 mites between 1st of December and the last day of February. And that is based on a wash of 300 bees. But the question often is like, 1 or 2 mites, 3 or 5 mites in an alcohol wash might not seem like a lot of mites that we are dealing with, but I just want to walk you through a calculation, and please excuse maths late in the evening, but just to sort of understand a little bit more closely, what does that actually relate to in terms of the number of mites that we might be dealing with inside of a colony. So first, it is not an easy question to answer, because it depends on a whole lot of things, and that includes the amount of brood that is present within the colony. It also depends on whether we're dealing with drone brood versus worker brood, what time of the year is it, are the bees still actively breeding or not, what climate are we dealing with, is the colony broodless or not, and also the level of infestation. But in saying that, if we just assume that we have 6 mites per alcohol wash, so per 300 bees, that would bring us to a 2% infestation rate. If we are dealing with, let's say, a winter colony or a small colony of 20,000 bees, which is roughly 2 kilos of bees, and if we also assume that we have 2 frames of capped brood, so probably we're dealing with a colony in wintertime closer to the coast, where we're still experiencing brood. And now we want to also know what does that relate to in terms of the mites on bees. So if we have a 2% infestation, we're dealing with 20,000 bees. That means we have 400 mites on adult bees. Now we not only have adult bees in this colony, but we also have the brood. And this is now a big assumption. But if we are saying that also 2% of the brood is infested, which is very conservative, because there's definitely going to be more mites, but let's stick with the 2%. So we have 1 full depth brood frame, roughly, or on average, has 6,552 worker cells. So if we say 75% of that brood frame might be covered with brood, we always have a little bit of honey, a little bit of pollen. We are now dealing with 4,914 cells. So 2% of those cells brings us to roughly 98 infested cells as the very minimum. Now we said we have 2 frames, so that makes it 196 mites in the brood. And keeping in mind from early on, we were saying that we have 1 to 2 mites emerging from a worker cell, we have 2 to 3 mites emerging from a drone brood cell, plus the mother mite, which potentially could reproduce again. So on average, and also again being conservative here, if we times the 196 mites by 2.5, just on average across the number of mites that we might find in one cell, we end up with 490 mites. So by now, with the bees, with mites that are reproducing in a brood, from a 2% infestation rate and 6 mites in an alcohol wash, in this scenario, we are actually talking about just under 900 mites that are actually present in that colony. So suddenly it's becoming a much larger number from our only few mites that we might be seeing in an alcohol wash. If we now consider that in summer, this colony will have at least 5,000 bees, and could easily have between 6 to 7 frames of brood, you can do the calculations yourself and see how many thousands of mites that will actually relate to. So record keeping becomes an absolute must in not only has it been important up to now, but more so even now, that we can keep track of when have we last been monitoring for mites, what were our alcohol wash results, and what kind of treatment have we been using last, if in states where we already have to treat for mites. Because we want to make sure that we are not using the same chemicals of the same mode of action. So that just means the same chemical class, which could lead to resistance. So the key takeaways from today are really that mites are elusive. They're very easily hiding on the underside of honeybees. Mites allow the spread of viruses. We now rely on treatments for Varroa. I didn't go into much detail today, but whether they might be organic or synthetic, in fact, any treatment is stressful for honeybees. And if we have fed bees or well-fed bees, they can handle mites, viruses, and treatments much better than if bees suffer from malnutrition. And for that matter, also active monitoring, record keeping, and managing of mite numbers is key. Because at the end of the day, more mites just means a bigger damage on the colony. Some helpful resources that you might want to look up. So there's a couple of books. There's one on the control of Varroa, a guide for New Zealand beekeepers by Goodwin and Taylor. There's also a document that can be downloaded from the AgriFutures website for free on resilient beekeeping in the face of Varroa. We've got several fact sheets and videos that are available either through the ARBIC website, the YouTube channel, or also the New South Wales DPI website. And definitely keep an eye out for the upcoming free workshops that are run on Varroa and in person. Don't forget about the Varroa development offices in your states. And there's always the exotic plant pest hotline that you can contact and utilize as well. And with that, thank you for listening. And yeah, there might be some questions later.
Awesome. Thank you very much, Madeline. That was extremely informative. We've had some really amazing questions coming through, so we're going to go through a couple of them now, but I'll give you a little break. I'm going to shoot across to Tamara Prentice if I can ask the first question of Tamara that's coming from the audience today. Given the pollination event and the total number of hives needed, is it realistic to expect commercial operators to comply with the conditions of having no Varroa and having had a recent inspection?
Yeah, so look, the permit requirements that I had on the screen, and I should have explained this during the presentation, were just for Victoria. So that's for any people going into Victoria. So there's a difference there between when you've got come from a Varroa infested area and where you haven't. Almonds are 100% reliant on pollination. So bees will be moving into Victoria and even into the New South Wales pollination areas, almond pollination areas from Queensland. But also New South Wales-based beekeepers can apply for New South Wales almond pollination contracts, regardless of their historic Varroa status. The Victorian government are realistic that Varroa is likely to be detected in Victoria in the next season. But pollination is not the only reason that border discussions between the jurisdictions have been required. The honeybee industry also relies on honey production across borders, queen bees and hive products as well. All of this has been really carefully considered and it aims to balance out the risks. We've been reliant on the honesty of all the beekeepers throughout the entire response. And we continue to share as many information and as much information and as many reasons as possible as to why collaboration and that honesty is so important going forward now for this industry. So.
Yeah, great. Thank you. Now I'm going to hit Madeline with a couple of queries that have come in. So if you're ready, Madeline, we wanted to cover up just a couple of queries that have come in, and there's quite a few that relate to monitoring and mite biology, obviously. So I'm going to try and put a couple to you at the moment. So the first one is, could you tell us how important it is to sample from uncapped versus mostly capped brood, which are well covered by nurse brood?
Yeah, very good question. And it is important to choose the right frame because if you think about, we were talking about the life cycle earlier, and the mites like to enter the brood just before it is sealed. So that is the time when the Varroa will jump into the cells and when the Varroa will be present on the nurse bees that are feeding that larvae. So we want to choose a frame that is just in that stage of being uncapped or just about to be capped, because that's where you will find the Varroa on the nurse bees.
Okay, and while we're in this space, given the foundress mite lays a male mite first, and that mite mates with subsequent female mites, is there any possibility of a biological blocker that would prevent the foundress mite from laying a fertile male mite? Would this then stop that cycle of Varroa reproduction?
I mean, if that was present, then for sure that will stop the mites from breeding with each other, right? I don't believe we are quite there yet, but that could certainly be one way of being to break that breeding cycle as such.
Yeah, yeah.
But it would
okay.
have to be very effective in terms of like working very efficiently, because as soon as you have one male that still makes it, you start the whole cycle again. Like you might slow it down, but yeah, I would imagine it would have to be really like almost 100% effective for that to really reduce the or stop.
Very novel technology.
Yeah.
The insect world is such a complex reproduction. So I get you there. I think the level that you went to has given people some really kind of great level of thinking to give you queries. So one of the other parts I just wanted to briefly touch on that was a great question too was, and I do know there's some answers to this one. So are there any known natural predators that feed on mites that we should be looking at?
There is. So there is a pseudoscorpion that actually feeds on the mites or attacks the mites. But with biological controls, it's always difficult because well, it depends on what situation. But in this case, like it would be hard to actually be able to have that biological control on a commercial scale or even on a level where it would be able to control Varroa. So it's by no means effective enough to be able to act on its own, if you know what I mean. So there's always the best way to approach Varroa is from an integrative pest management approach, where you use different ways and different techniques to control Varroa. But biological controls on their own are not effective enough to control Varroa, unfortunately.
Yeah. Okay. Agree. Great info. I just wanted to touch on the advice to check your hives every 4 weeks. That, once we've got Varroa, we're finding it as a really key critical component now to changing the messaging. And this might not be true for a little while in some places, but once we've had Varroa that's been infesting hives and have reinfestations, we do have new messaging coming out that's saying, don't wait for 16 weeks, get back in and do this as soon as you can. So there's 2 questions I'd like to put to you around that. We're telling people to monitor every 4 weeks once they have mites present, but could you advise if testing should continue during treatment? If someone's using like a behavioral treatment, for example, for the 8-week cycle, should they test halfway through or once the treatment's finished?
I mean, it definitely doesn't hurt, but whether you really need to do that is, I think that's more up to you. But you definitely want to test 2 very critical points, and that is before you put the treatment in, so you know what level you're dealing with, what is your threshold or what are the mite counts, and you want to test straight afterwards so that you know how effective that treatment was.
Yeah. And there are some treatments that you'll need to read the permit and the label to make sure that
that's right.
It's clear to you that you shouldn't open the hive during some of them. So be very clear on what product you're using. One other part around that was you mentioned Varroa mite treatment not tailored for the Australian conditions or the environment. did you want to elaborate on why that's the case?
With treatments, or with how the mites actually like increase through seasonal?
I was early on. It came in quite early in your presentation. So yeah, the question is why Varroa mite treatment's not tailored for the Australian conditions or the environment.
Well, it depends which treatment that you might be looking at, but there's some treatments that have temperature restrictions. So, for example, formic acid or formic pro, also Epigard, they have restrictions as to when you can use them or when they will be effective. So that is between, like, for example, for formic pro, between 10 and 29.5 degrees. Just because once it gets too hot, it will release too quickly, or if it is too cold, it won't be effective enough. So that's where it become a little bit of a challenge in Australian conditions, because we do have very hot temperatures. And yeah, is there anything else that you can think of, Bianca, or did I answer that question?
I think that's great. I think it's just really to cover that we're using treatments that have been developed overseas because we've never had to go to that extreme in Australia. And so now that we're realizing that there are environmental limitations to everything that we're doing, and especially the cycle of our brood is going to change, and the treatment options that we pick are going to ultimately be determined by those environmental and hive conditions.
Yeah.
I think that's something I would be thinking of when that question comes to mind. But
yeah, and certainly also, I mean, we do have it very challenging with honeybees producing so much honey throughout the year. So that's another limitation as to what treatments can we use with honey supers on, with honey supers off, and hopefully we will have more treatment options available in future as well.
Yeah. Another question is, people have been told not to open hives when the temperature is less than 18 degrees so that the brood isn't damaged. And now that it's recommended we're testing monthly or every 5 weeks, say, is there a temperature limit when beekeepers should definitely not open hives and test for Varroa?
I mean, temperature certainly can have a negative impact on honeybees, and that is true for too cold and too hot conditions. So it goes both ways, but it also depends on how long that colony has been open for. Like I can't really give you an exact degree. You shouldn't be opening a hive if it is below 5 degrees or anything like that. It also depends a little bit on the health status of that colony. Whether we're talking about a really weak colony will be much more affected than a colony that is strong and healthy. But yeah, the critical thing that I would say is those really cold temperatures and also really hot temperatures, and also be considerate of how long a colony is actually open for, and try and minimize the amount of time that you actually spend in disrupting the colony.
Yeah.
Not disrupted for more than what is actually necessary.
Yeah. Okay, cool. A great question in relation to cold is that beekeepers have noticed from the heat map that the coastal areas seem to be mostly affected with Varroa. Is there anything about the Blue Mountains that is helping slow the spread?
Definitely. I would expect it in the Blue Mountains to be much colder this time of the year, especially during winter. So if you are in cooler environments and where the bees specifically might be experiencing a brood break, which could also be the case in Tasmania, for example, or further inland, then you definitely have an advantage, because during those periods of time, the mites will not be actively reproducing. You might still have mites eventually present on adult bees, but they will not be reproducing.
Okay. One of the questions that's come in is in relation to the heat map, that the heat map is not showing some stories coming out of industry that, and this might be a tomorrow question, but the heat map isn't showing some of the information that might be passed around industry that there's different areas where Varroa is present. Did somebody want to touch on the confidence we have in the heat map and the amount of reports and the importance of reporting whether you do or don't have Varroa to make sure that that heat map is accurate as we can get it?
Yeah, I'm happy to do that, Bianca. Look, as I've mentioned a couple of times through the presentation, the heat map really does rely on the accuracy of reports. So, for example, we had a report west of Canberra a couple of weeks ago, and it was a new area. And in New South Wales, you know, we aren't still checking reports, but where we are getting outliers, if we have got people able to go, we are asking people to send us photos, or we send videos out to check what's going on. And so that one west of Canberra, we didn't publish, and we went and met with the beekeeper, and it was found to be pollen mite. So that one didn't get published on the heat map. In areas where there are known infestations, we're not checking. And similarly, we're not checking negative surveillance responses. So the accuracy of the heat map really does depend on the reports that are being submitted and people being trained up in what to look for in terms of Varroa before they're doing those reports. So please do the training, come along and do the formal training or the TOCAL online training, and then submit your reports. But yeah, there is, I mean, some of the data is, you know, it does depend on the knowledge of the beekeeper making those reports. But we'd like to think that most people are pretty good. So we'd like to hope that it's got a good level of accuracy. And particularly the heat map is designed so that the more positive reports you get, the stronger the red, the more negative, the stronger the blue. So if you're seeing repeated hot detections, well, then it's likely a high area of Varroa, whereas you've just got a really light red one detection, then it could well be a misnomer. So it's just something to be wary of.
Yep. And I've heard from a couple of different people that there's reports of Varroa in the Dubbo region, but until that heat map receives a positive report from the industry, it's up to beekeepers to be able to look at that map and feel confident as well that the industry is doing the right thing to whether it is or isn't out there. And generally, when that happens in other areas such as Nowra, as recent as 2 weeks and a week ago, we're sending videos into that region and we're getting beekeepers calling up for some help. So it's actually in your best interest, because it's a great way to navigate the assistance and the training and that kind of thing towards the places where it's needed the most. I'll move to another question that I might bring Liz in. Elizabeth Frost, Technical Specialist Honeybees, is in the background as well, working with us this evening. We've got a query about why the threshold that we're producing out of the program at the moment is lower than the US thresholds, which is usually 1 mite in an alcohol wash. Could you talk to those points, Liz, about why we're using a higher mite number in our alcohol wash per 300 bees?
Yeah, definitely. So important to note that we have Varroa, but we don't have deform wing virus detected yet in Australia. And deform wing virus really changed the game in the US in terms of what threshold levels were acceptable to maintain, beekeeping for leisure, backyard honey, commercial business, sideline business. So the levels in US commercial industry are the same as what as what we're recommending here in Australia. And those numbers are still published in a current document that's taught across North America, in Canada and the US, come out from the Honeybee Health Coalition, which I'd encourage everyone to read. Obviously, it's not got the similar, it's got most of all the treatments that we have here, but some that are not legal here. So that component's not applicable in Australia fully, but the threshold is. So we've currently got the, I guess, quite liberal threshold present in Australia, which some beekeepers are like, oh, why is it so low? And others are like, why is it so high? But essentially, it's a number that may change over time as we understand more about Varroa in the Australian environment. And right now, we've got some questions about the 2% threshold and do I need to treat immediately? Right now, it's not a mandatory treatment, it's highly recommended. So you might say, I've got my backyard hive, I've just done my alcohol wash, I've got a 2% infestation, it's winter, can it wait a little bit? And the answer, frustratingly, is it really depends on where you're located, what the reinfestation pressure is. So if you wanted to, act with an abundance of caution and you had a 2% infestation right now in your hive, I would recommend treating because I don't know the reinfestation in your zone, and it could be severe, particularly if you're on the coastal fringe, like we're seeing on the heat map. Is pretty hot with people actively monitoring and reporting, so we can all see.
Yes, yeah, for sure, absolutely. I've got a lovely, wonderful question that I'm going to pitch across to Madeline. So thank you, Liz. Trying to do this as smoothly as I can. Could we ask Madeline this query? Can you supply your hive with pollen that you buy from a beekeeping supplier?
Yes, you can. So there's pollen available commercially to buy for beekeepers. And as far as I know, though, they can only do that in WA and in New South Wales, but that doesn't mean that you can buy it from a different state, or that you cannot buy it from a different state. But there are also other pollen supplements available, and there's quite a few of them available on the market. So definitely your local supply shop will be able to help you out with one or the other.
Okay, very good. Another query around the deep dive part of your presentation, really cool images of the slices of bees. Just have to say, I thought that was really amazing. This query is regarding the way that mites feed on fat body. Is anyone looking at developing a liquid pesticide that we could feed our bees in a sugar syrup that could kill the mite and therefore break the cycle? I have seen a bit going around this, not necessarily a pesticide, but fungi. But I'll let you answer.
I mean, it is a novel way of thinking about it, because that way you would affect like whatever the mites are feeding on the bees, and then as a consequence, they somehow getting affected by that because of what they're getting from the fat bodies that we have been feeding to the bees, I guess is the concept. But yeah, I haven't actually seen anything as such that has really been tested or effective way of doing that at this point in time. I think it is an interesting idea. Whether it may or may not be something that we might be able to do in the future, who knows? But yeah, it's in some ways Varroa are quite simple creatures and also very sophisticated. So yeah, it's always more complex than we think. But yeah, at this point we need to consider everything we have at hand to be able to control that beast.
Yeah.
So yeah.
Back to feeding. Is it safe and recommended as okay to feed during bees during the winter when there's a Varroa treatment in the hive?
Whether feeding during winter may be necessary or not really depends on where you are. So it doesn't necessarily make sense to generally feed bees during winter time. Like it depends what your purpose is or what you're trying to achieve. Like, are you feeding because you're still on coastal conditions or somewhere further north where it is warmer, or you're trying to feed bees to build them up in preparation for pollination? So I think the first question to answer is really, why are you feeding those hives? And then depending on what you're treating with, like let's say if you've got strips in your hives, like there's no problem in terms of still being like feeding colonies as such, but it really depends on also what you're treating with, how you're feeding. Like for example, if you're treating with formic acid, you don't want to be opening the hive halfway through to putting a pollen paddy, or to put in a sugar syrup in a frame feeder or things like that. So it does depend a little bit on what you're treating with, and yeah, but keep in mind why and what for you're feeding hives at a certain time, or whether that is winter, spring, summer feeding.
Yeah. And I note someone has asked about feeding bees pollen and the requirement and best practice for that pollen to be irradiated. Did you want to touch on that as a prevention of other pest and disease transmission through colonies?
Yep. So irradiation of pollen is a must. So whether you are trying to trap your own pollen and want to feed it back to the bees, or if you're buying pollen from a supplier, it will 100% be irradiated. Reason being that pollen is a way of transferring fungal spores, or whether it could be chalkroot, could be nosema, also AFB spores can be spread that way. So definitely needs to be irradiated.
Lovely, thank you.
Thanks.
I've got a query across to Liz. For this one is for our Varroa treatment plan. Has there been any research on treatment cycles complementing each other? So Baverol followed by Formic Pro, or having a better outcome with Baverol followed by Apivar, or is it just to the individual beekeeper to decide their preferred treatment cycles?
Yeah,
really good question.
Complicated question. Ultimately, it is up to the beekeeper to make the best decision for their bees, their environment, and their scale of beekeeping. And I guess there isn't one recipe that is the best, the best sequence for all. It will really depend on your environment and what your hive is doing at that time. So the temperature, the environmental temperature. Obviously, we know a healthy beehive maintains its own temperature, but those temperature restrictions on the chemical are about environmental temperature. What are your daily highs and lows? As well as what's your hive doing honey-wise? If it's making honey, you have 2 treatment options available to you, Baverol and Formic Pro. If it's not making honey, you have the other 4 options. The Apis, Apivar, Apitraz, Apistan, and Formic. Sorry, help me out there, Bianca. We got 4 there. I just was typing a response to one of the folks, and I'm getting my.
I'm actually reading the next question.
So I suppose the one thing I could say that is a recommendation that other countries have looked at for beekeepers to think about in their treatment plan is to talk to those beekeepers that are in your immediate area. The best thing you can do is to be monitoring and talking to each other about your Varroa levels and when you need to treat and what you're treating with, and understanding for your treatment plan what those mode of actions are for those chemicals that you're using. So we don't want to use chemicals back to back that have the same mode of action. So for example, I'm not going to use Baverol twice in a row or Baverol followed by Apistan. I'll need to follow that with a different treatment that has a different mode of action, such as Baverol followed by Formic Pro, or Baverol followed by Apigard, or Baverol followed by Apivar, Apitraz. Get your flashcards out and look at the Varroa resources that we've got on the website to get into your head, what rotation that you might have available depending on your environment as well.
Yeah, get to a training event, get to those workshops, start learning now. Yes, it is a long road ahead of figuring out what will work for individuals in the way they do bees. I think it's going to be a lot to take on board, a whole new level of learning for everyone. Another query that I thought I might ask you about was novel treatment in terms of heating hives. This one you have answered in the chat, but I wanted to put this into the Q&A so that it's there for later. The query was, has anyone tried using a heating mat to keep the hive warm during winter, if and when you have to inspect the hive?
Yes, I think that was about, okay, gotcha. Yeah, so heating mat to get your hive a bit cozier for when you disturb it in Australia's winter months. So I guess to put it in perspective on Australia, winter, you guys define it a bit differently than we do in North America, where it's really, really cold and a lot of it snows. So I would say the key thing to remember for Australia is to understand your mite levels in autumn. So at that time period when your hive is developing its overwintering bees. So those bees, you need to protect them because they're going to keep your hive going through winter, or you need that big population to go into, say, almond pollination or canola honey production, early season seed canola or honey canola. So the key thing is to understand your mite levels in autumn and winter and control as needed, so that when that hive might suffer from cold stress, if you had to test for mites during winter, the dead of winter, you could have potentially avoided it, I suppose. So yeah, you have to be a bit flexible. If you're in an area that you think you'll have high mite infestation, you've done your early winter mite monitoring event, you got 2% infestation, you treat, you wait 8 weeks or whatever the treatment time frame is and remove the treatment. You do another monitoring event, say at that point, to check that your treatment has worked. For curiosity's sake, you might do another one during a nice weather window. And if that level is at 2% again, then it would have, obviously this is a calendar year ahead of us where you do have mites now to keep really good records. So you can kind of get an understanding of an environmental pattern as we get Varroa expanding in the environment.
Wonderful. Thanks, Liz. I just wanted to touch back to Tamara with a query that came in about not updating maps in areas where existing infestations are occurring. And that, I think there's been a misinterpretation of that particular listener. Tamara, can you please explain? Like, I know that the heat map is continually being updated on a weekly basis. Could you talk about the progressive amount of times that that's being updated, our cool, hot heat map?
Yeah.
And also around the fact that if there are new infestations that are reported to the heat map, that the team within the program makes a decision whether that's an outlying new sort of area that never had Varroa before, and what kind of contact the program is providing to give us some understanding. Like you explained the incorrect or suspect mite, which was a pollen mite, that there are other areas like Nowra and Bega where we've had BDOs go out. Could you have a chat about that process and a little bit about the updating of the actual heat map?
Yeah, sure. So I'm sorry I wasn't clear earlier. So the map is actually updated whenever a positive report is submitted, and it just deepens the heat in an area where there is an existing incursion. What I was meaning when I was saying that is that we don't go out and do inspections in those areas routinely, because if someone submits a positive Varroa report in an area where we know we have Varroa, it's likely that it is Varroa, and it's not going to mean a difference in terms of treatment and monitoring and looking after hives for that particular area, because you're already in the midst of a response. Where we're getting outliers, and they were the ones I was talking about earlier, that's where we do have, when we can, our videos going out and helping those local areas to prepare as they start to fight the earlier stages of that incursion. So we're increasingly trying to then contact local bee clubs in an area where we're seeing a response, and offering to help people on the ground in those circumstances. So if you're not a member of your local bee club, join up, and certainly subscribe to the EDM. We've put the link in a couple of questions and answers in the chat. So subscribe, because we do talk in our weekly updates about where we're seeing new areas of incursions. So that's a way to keep up to date if you don't want to go check the weekly, the heat map on a weekly basis. So it's updated on Fridays at lunch. It will continue to be updated on a weekly basis. Once it goes national, that's still the plan as well. So yeah, keep an eye on that. But otherwise, subscribe to the weekly updates. You'll get information of where the spread's up to, as well as a heap of really cool information about the things that the program's offering, and training in your area, and whatever other findings we might come across as our response to this starts to change as we continue to learn how to manage living with Varroa. Thanks, Bianca.
I think that brings us to an end for this evening's webinar. I would love to just thank everybody for tuning in, and for all of the fantastic questions that have been put through. A thank you to our team behind the scenes, Joanne Cumming, Ellie McNamara, Sheila Stokes, I know is in the background, Elizabeth Frost for joining us and bringing forward some really fabulous answers. and obviously our presenters, Tamara Prentice and Dr. Madeline Kratz. A great big thank you to our community of beekeepers across Australia for tuning in and listening, and especially if you're listening back later on. This webinar is recorded and will be available in the coming days, so keep a close eye out for some details to come. And good evening. Thank you.